Good morning, everybody. Welcome to the last day of the workshop. It's a great pleasure to start this morning with Ultan McDermott, who was a group leader at the Sanger Institute for a long time and is now the, what's the official title? You're the Chief Scientist Oncology at AstraZeneca, and is currently our colleague at CI, where they are still hosted while they build their big new building right next door. And he will tell us about pharmacogenomics.
Thanks, Florian. Thanks for the invite. So as Florian said, I wear a few different hats. I still practice as a medical oncologist for 1 day a week, treating patients with colon cancer. Been in academia. I did a spin-out company. And then I've gone to the dark side. I've joined a pharmaceutical company, AstraZeneca, which moved down to Cambridge from Manchester probably 6 or 7 years ago. Really because life's too short and it's about trying different things out. It's been quite a lot of fun, actually. I suddenly realized that what I would have viewed as job done on the academic side is job sort of 20% done when you get into making a drug for patients. There's different sort of pressures, but there's actually quite a lot of interesting science. And increasingly, I would say, certainly for my younger colleagues, there's lots of things you can do in life, and science as a background, and mathematics and chemistry, the life sciences is a wonderful foundation to do whatever you want to do. So when I grew up, certainly coming from the medical side, you did one thing. Or if you're in science, you had a very career path, you became a PhD, you did a postdoc, team leader. I think it's a lot more fluid now, and it's about finding science that interests you and following that wherever it leads you. And it might lead you in lots of fun and interesting places. The world is a small place. I'm going to talk about pharmacogenomics. This has really changed quite a lot, and fundamentally it's about not treating every patient as though they were the same. And I think, as I'll show you, it's becoming quite clear with the advent of, over the last decade, next generation sequencing, that not only is a lung cancer different from a breast cancer, different from a melanoma cancer, but even within those cancers there are profound molecular differences. And you might say, as my old supervisor used to say, so what? But those molecular differences seem to have an impact in some cases with whether patients live or die, their survival. And more importantly, when we talk about pharmacogenomics, those differences have an impact on response to treatment. And so almost inevitably in most clinical trials now, we begin to think about how we stratify patients. That creates a huge complexity because instead of treating every lung cancer the same, you're now saying, well, in fact, it's not just the tissue type's important, the molecular subtype is important. And that means now we're targeting very small percentages of patients of different drugs. That creates its own economic and clinical problems as well, and how we actually detect those patients. And if you're looking for a molecular subtype, which is present, say, in 2% of lung cancer, you've got to screen quite a few hundred patients to get those 2 patients who have that molecular alteration, and that creates a pressure within the health system as well. So this is definitely not a presentation I would do as a whiteboard, by the way. Drawing NGS by whiteboard would be tricky. It's just the whole thing's frozen. Okay, so this is a rather morbid title. We all die of something, but the reason I got interested in cancer is it's highly prevalent, and as I said, probably 1/2 or 1/3 of us will get cancer during our lifetime. So it's a huge problem. It's not going to go away. It is a problem largely as we age, but it's highly prevalent. If you look at the most common causes of cancer death, you can see that despite the advances we have made, and the blue and pink is male and female, is things like lung, bowel, prostate, breast still kill a lot of people every year. And that's the reason we work in these cancers. So these are huge clinical problems. But what's interesting, as I said before, is we're beginning to realize that we should target different molecular subtypes in different ways. And for these cancers which have good survival and high prevalence, that's fine. But you can see there are a whole bunch of cancers which I've highlighted in the second box, which have really, really low survivals and are reasonably prevalent. And so certainly in the UK, some of these cancers have become the focus of Cancer Research UK's research strategy. Things like esophageal cancer, brain cancers, lung cancers, pancreatic cancer in particular. So this is a very famous set of pictures this was a melanoma patient from Boston. This was kind of the paradigm of personalized cancer therapy. So this is a patient who's got a skin cancer called melanoma. They're almost all, probably about 60–70% are driven by a mutation in a gene called BRAF. That's what's called an oncogene. These are genes that confer a selective advantage, that when this gene is mutated, these cells proliferate more rapidly. And what you can see in this patient is a number of different sort of lumps on the skin. These are essentially cancer deposits in this patient's skin. And you can see here that after treatment, and I had mentioned before, this tumor has an activating mutation in the BRAF gene, and this patient was treated with a small molecule inhibitor targeting that BRAF mutation. So this is a classical paradigm of personalized cancer therapy. You've got a tumor, you know the mutation, you target that with a drug which inhibits that, and you go from here to here. So you can see that a lot of these bumps have sort of largely regressed. You can still see a bit of them here. Patients put on weight. There's a whole separate discussion which really is beyond today, which is the fact, unfortunately, that for many patients, although we're doing this bit to this bit well, what happens over time as tumors evolve is you get drug resistance. And one can imagine in a purely Darwinian sense, you have a population of hundreds of millions of cancer cells. They all have some particular vulnerability. We target that now with the right drug. Maybe 99.999% of the cells do commit to cell death and die. But unfortunately, over time, there's a small percentage which eventually emerge. And whether they stochastically acquire a resistance mutation by chance, or there is a pre-existing minor event, it can be either. But over time, and it could be over a year, year and a half, the cancers in many cases does come back. It is now resistant. There's a whole interesting piece now on in between what happens here in the non-genetic resistance space. So what we've traditionally done is we've traditionally sequenced patients here. They've got the right mutation. We give them the right drug. They get resistant a year later. We rebiopsy the patient. You can actually see the rebiopsy site here. In this case, this patient's recurrent tumor has the original BRAF mutation, but it's now acquired a new mutation in a gene called NRAS, which is another oncogene in the same pathway. So the cancer has essentially acquired a new mutation, which keeps that original pathway now active despite the drug. And so we've been very good at doing that. There's a huge amount of interest in what happens in the middle here, because it's quite clear that by definition, this tumor's come back because although a lot of the tumor looks to have disappeared, there are still viable cells there. There must be, because the tumor has come back a year later. And with the advent of single-cell sequencing, we can now look at some of these viable cells in this middle stage and realize that there appear to be non-genetic mechanisms of resistance. Now, it's not well understood. It's a very hot topic in resistance at the moment, because one of the challenges is that if you wait until this happens, I've mentioned that this patient, who was a BRAF mutant melanoma, acquired an NRAS mutation, but the next patient might have acquired a KRAS mutation or a KRAS gene amplification. There's a huge amount of heterogeneity here in that the next 10 patients who develop resistance could have 10 different resistance events, and it gets even worse. Within that one patient, if I biopsy 10 different metastatic sites which are resistant, they don't all have to have the NRAS mutation. 4 of them could have the NRAS, 1 of them could have KRAS, and the other one could have something else. So there's heterogeneity between patients and also quite a lot of heterogeneity within the patient. And there's a lot of interest as to whether targeting the non-genetic resistance here might actually prevent this happening in the first place. But we don't know that yet, but it's a massive, massive question and hugely important. All of what I've talked about personalizing cancer therapy, pharmacogenomics has really been driven by next-gen sequencing. And you know, I come from a time pre-NGS, and increasingly there'll be less of us around, but there are most of us who remember what it was like for capillary sequencing in the old days, or just conventional PCR. But next-gen sequencing essentially has allowed us to look at sort of predominantly what's happening at the DNA level and also in RNA sequencing. I won't talk about proteomics because I think proteomics technologically has lagged somewhat behind, but it's really now coming up quite fast. I mean, you've always made the argument that ultimately it's proteins do the work, but it's a much more complex space to explore. But whenever people talk about next-gen sequencing, if you've never seen next-gen sequencing, this is next-gen sequencing. When people talk about I'm looking at 100 reads or 50 reads, I'm looking at my mutant reads, it all sounds quite complicated. But in essence, if you sit down at a genome browser, this is essentially a tumor. You're sequencing sort of forward and reverse, which are sort of the blues and the reds. This gene is BRAF, the one I talked about that is heavily mutated in this melanoma skin cancer. And you can see when you look at the tumor reads, so each of these lines is a read. This is the reference, and you can see here in red, the browser has highlighted a T2A mutation change. And this is the classical BRAF mutation that drives that melanoma skin cancer. So when people talk about looking at NGS, and if you've been at a kind of a large sequencing institute, this is what a lot of scientists look at, sort of this type of data. So it's not very complicated. But I think the big change is that really, certainly on the clinical side, up until recently, everything we've done when we see a patient has been very much based around the microscope. And arguably, from the 1600s, it probably has changed a bit, but it hasn't changed a huge amount. We do H&E stains, we look at tumours, and we look at how big they are and how aggressive they look, and are they involving different structures. But of course, if you buy into what I've said, that there's now heterogeneity and different subtypes within a cancer, then you realise that that's not sufficient. And so increasingly, what we're beginning to do is go towards a unified classification, where we're using now microscopy and immunohistochemistry, and that's still a hugely useful tool. But we're beginning to marry that with different types of omics. And that is very much still DNA omics when you look at the hospital level, because DNA is relatively stable. You can get it out of archived tissue. Transcriptional data is still not used that widely in most clinical practice. Whether that'll change, proteomics, definitely not, much less so. But there's certainly, for most tumour boards, a routine discussion of mutations, copy number alterations, together with the histology to try to guide how we treat patients. And what's driven that whole subclassification, that every lung cancer is different, every breast cancer may be different, is really endeavours like this. So probably about a decade ago, the Cancer Genome Atlas, which is represented here, and the International Cancer Genome Consortium, these were large international endeavours which said, look, we know there's probably differences, molecular differences within cancers. Let's find out what they are across thousands upon thousands of genomes, and let's make that publicly available. And again, as me, if you were pre-TCGA, for some of you guys, going to see Bioportal is, take it for granted. I'm just going to go to see Bioportal. I'm interested in the prevalence of my favourite gene and favourite cancer. You guys can just go now to see Bioportal and do that instantly. You should imagine what it was like before that. You couldn't do that. It was quite hard to get that information. So I think these are great examples of how, working collectively and putting data into public domain, you can really empower. And those sort of resources have really empowered our ability to look across all of these different cancers and realize that there are different molecular subtypes between cancers and also within cancers. And so routinely, within many cancers now, we think of not particularly whether it's a melanoma, but is it a BRAF mutant melanoma? Is it an NRAS mutant melanoma? Is it a non-BRAF, non-NRAS? In the setting of lung adenocarcinoma, which is a common cause of lung cancer, we're interested in whether it's KRAS mutant, EGFR mutant, ALK-RET. So that molecular classification, we take it for granted now, but there was a time when it didn't exist. And it largely didn't exist because we didn't have these comprehensive evaluations of essentially cancer genomes across, now tens of thousands of genomes. And this is a sort of an example of what this data looks like. So this is an example of, probably a couple of hundred melanoma skin cancers. So essentially, each of these columns is an individual patient who's had his or her genome sequenced. And you can see highlighted in the coloured boxes the different types of mutations, whether they're hotspots or not. And so you begin to see patterns, for example, about 60% of the BRAF mutation, about 30% are NRAS, and then there's a small proportion which are double negative. And then you see the other mutations. And so for the first time, getting a glimpse of this landscape allowed you to ask, well, should I be treating these guys different to these guys? And even more profoundly, within the BRAF mutant population, do these additional co-mutations have an effect? Now, that co-mutation effect becomes quite complicated because we're, in many cases, underpowered to really dig into that in a substantive way. And we quite often don't have the biological models to let us get into that. But that's a big question now. Ooh.
Was that resolution battery exploding?
Oh, you okay?
Yeah. I'll just take it outside. Yeah. And then buy a new battery. So what if
Oh, wow. So it does happen. Okay.
But we're not planning special effects for all talks.
That's right. So the takeaway message is, this is one cancer. You can see, if you believe that these mutational events have biological function, there are obviously differences. And even within the very common subtypes, for example, the BRAF mutant melanoma, which is the one you read about most, you can see here that some are P3 mutants, some are P10, CDKN2A. Do these additional co-mutations have an effect? And increasingly, that's going to colour how we treat patients. Now, The challenge clinically is you're now dealing with smaller and smaller and smaller patient populations, which means now your clinical trials have to get bigger and bigger and bigger because you've got to screen more and more patients. Because if I was looking at a drug that I think works in BRAF mutant melanoma, and where they have additionally P10 loss, that's 7 or 8 patients out of a couple of hundred, instead of traditionally just saying, well, all of the BRAF mutant melanomas are 60%. So these are just clinical challenges for us as we try to design these trials. But it's an unprecedented view. It's exciting, points to different biology, different ways of treating patients, and there is no one size fits all. And equally, this is the same in lung cancer. So you can see in lung cancer, a very large number of P53 mutations, but then you have these other co-mutation events. And you begin to see, obviously, as well as the fact that there are different groups of patients, you begin to see these interesting stepwise patterns. And so those of you who do informatics, we call this mutual exclusivity. You see genes that never appear to be mutated. So if you imagine each column as a patient, you can see that the CDKN2A mutant patients don't overlap with the P10, who don't overlap with these guys. They're never mutated together. And that may tell you about biology, because, of course, what you may begin to realize is that some of these genes that are never co-mutated, it's essentially because they're in the same pathway. And so genomics, static DNA, that sort of imprint of tumorigenesis can actually tell us quite a lot about biological pathways as well. You need big numbers to do this. So I'll talk a little bit in a sort of high level about what are the genomic alterations clinically we look at whenever we're thinking about how we stratify patients for treatment. And broadly speaking, we look at essentially mutations. We call these either substitutions or point mutations, where one letter switches to another, or we talk about what we call insertions, deletions, indels, and it's where you get either big deletions or insertions of DNA sequence, and they can affect gene function. Gene amplifications are where you get essentially multiple copies of a gene, maybe sort of tandem duplications or double units. You can get gene fusions, where you get 2 genes which break apart and stick together. And in that process, one of them now becomes typically overactive and drives cancer. And then there's a whole, I shouldn't say industry, would be the wrong word. There's a whole interest now in what's called mutational signatures. So these first 3 are quite obvious events where we're looking at genes and whether genes are abnormal in some way, and whether that abnormality says I should treat you differently. There's another way of looking at genomes, which is much higher level, which is, forget about gene level, you're looking at just the pattern of all these hundreds of thousands of mutations across the genome. And the genome's, about 5 billion bases wide. And that pattern of mutations and where they lie, and which of the 4 different nucleotides they affect, gives a kind of a signature. We call these mutational signatures. I'll show a couple of these, because these, I think, will become increasingly important. So again, when we talk about what these mutations look like, if you're looking at NGS data, I mentioned before that, if you sequence a tumor, you get a whole bunch of reads, forward and reverse, and then the browser will typically compare to the reference genome, highlight in red, where's the mutations. This is what a BRAF mutation would look like. And again, if you look at an indel, so this is a deletion. It's a sort of a 14 base per deletion, and you can see here what will happen is it'll show you that this bit of DNA is essentially missing. It's been cut out in that gene. This one letter changing to another nucleotide letter activates BRAF. It's an activating mutation. This deletion equally, because it's in frame, that also activates, in this case, a gene called EGFR, and that's a very commonly mutated gene in lung cancer. And importantly, both of these, if they're present, open up the possibility of targeting treatment in that patient with a very specific therapy. This is got skewed a little bit, but this is a good example of the power of pharmacogenomics. This was a study carried out quite a few years ago now, 2005, almost pre-genomics, treating essentially lung cancer patients, so quite a few thousand lung cancer patients with a drug called gefitinib, which essentially is a small molecule inhibitor of that gene EGFR. So this is a targeted therapy versus nothing. And they looked at it, and we used to joke as oncologists that if you can get your laser pointer in between the 2 survival lines, there's a bit of a benefit. That's how bad oncology used to be. We didn't ask for very much, but you would look at this, and even if I look at the p-value, the new drug in red isn't doing very much compared to placebo. You'd sort of say that's a bust. So this was a trial, many, many millions of dollars to run, no benefit. But of course, what happened, unfortunately, it did happen retrospectively, is they looked at these patients, they said, well, you know, if we look at these lung cancer patients and we realize now, because genomics was now beginning to appear, that there may be a subgroup in here that actually do benefit, then of course what you see is this, which is that if you now identify the 20% of these patients from the red group who actually have a mutation in EGFR, they do much better. Their response rate was 40% versus about 4% for the unselected. So you target the right patient and the right mutation of the right drug, you do get a benefit, but you've got to know what that right molecular subtype is. And so the following trial, a couple of years later, again confirmed that if you take EGFR mutant lung cancer patients and you treat them with this targeted agent versus standard chemo, they do much better. You can easily fit my laser pointer between the two lines. Now, the more astute of you will realize that that's great. These patients do seem to, you know, survive, but everyone eventually dies. Because of course, we're not going to talk about it today, with treatment over a year, year and a half, you eventually almost inevitably get resistance because cancers are constantly evolving. And there's a whole realm of this non-genetic resistance. And that's the next big challenge, actually, is what we do here when that cancer now comes back. Because your first drug doesn't work anymore. Cancer's found a way around that. There are ways you can, things like CRISPR technologies and single-cell sequencing, we can probably predict what cancers do to get around that. And that's probably going to be the next wave of therapeutics. And it may be much more in metabolic space or epigenetic space. But you can get quite dramatic responses. So if you take an EGFR mutant lung cancer patient, and this is an EGFR mutant lung cancer, this is what's called a PET scan. PET scan is essentially radioactive sugar. Cancers light up nice and bright. EGFR mutant patient, you treat with an EGFR inhibitor, you go from that to that. So you haven't cured this patient because, you know, the tumor's still there. And, you know, one of the challenges is that over maybe a year, that will eventually get bigger and bigger. It'll eventually find a way around the original therapy. There's a lot of interest now in biopsying these very few cells in here and using things like single-cell sequencing to understand what are the non-genetic mechanisms that these cells use to survive, to tolerate treatment. It's kind of it's a gray area at the moment. It's challenging to do clinically because getting a needle into that to get tissue would be relatively straightforward. Getting a needle into that to get tissue is a bit trickier. Difficult, but not impossible, and it's such an important question that we will move in this direction pretty quickly. It has to be done. We have to know what's happening here, because if we wait now a year later, when this now goes back to that, it's now very heterogeneous. It may be multiple different mechanisms, and it's much more difficult to treat. So the premise is whether, if we can understand what enables these few cells to survive, and we can wipe these cells out, does that point towards a rational drug combination where you would treat initially to get from here to here, and then you have a second treatment which targets how these cells then survive. But of course, what these cells do, even if it's a non-genetic and altered transcriptional programs, that in itself could be quite heterogeneous. That's the big worry, is that, you know, cancer cells are pretty clever. They, you know, is there just one mechanism here that's enabling these cells to survive, and if I would add in one drug? I think it's probably unlikely. I'm not, you know, you need to be naive to think that. We also talk about gene amplifications. So this is typically where you duplicate a gene. So you have a gene which, you know, normally drives proliferation and cell division. So you'd imagine if a cancer has, you know, 40 copies of that gene now, it's now driving proliferation really fast. It's now dividing much faster. And so, you know, you can typically get, you know, tandem duplications, which is the commonest way. And if you look at a kind of a classical array plot, you'll typically see these as spikes like this, where you get an amplification. So this is an amplification of that gene EGFR, which again, as an oncogene, is very commonly amplified. And that's, you know, what it looks like. And again, you know, there's been interest really led by breast cancer, where there's an oncogene called HER2 or ERB2. It's amplified in quite a decent number of patients. And essentially, if you target, you know, this amplified gene HER2, with essentially this drug trastuzumab, which amplifies that gene, then you get, you know, you get this sort of level of increased response, and then you can get quite sophisticated and start really putting together the survival curve. So if you're a HER2 amplified breast cancer these days, you will get a HER2 targeted therapy now. And I'll show you towards the end. If you look year by year, you can really see with the FDA, which is the US organization that approves drugs, the big explosion in how many drugs now are underpinned by a genomic marker. There's been quite a big increase in those. There's another condition called gene fusion. So again, this is where you get two genes, but unfortunately you get a situation where they break apart, and essentially if they get stuck together again, and I'm showing this fusion here, it's a gene called EML4, which I think it's not quite clear what EML4 does, probably not very much. ALK is a tyrosine kinase, which has a role in proliferation and cell survival. Separately, they behave themselves, but when you get this gene fusion where they break apart and come together, where part of the fusion is EML4 and part is ALK, this EML4 ALK gene fusion essentially becomes oncogenic, becomes proliferative, essentially sort of helps form a certain type, and it's present in probably about 3 to 4% of lung cancers. So this was quite an interesting example where we went from a scenario where a lot of the certainly pharmaceutical companies weren't really interested in going after a molecular subtype if it was very low prevalence. That HER2 amplified breast, that's quite a decent chunk of patients, that's okay. The EGFR mutant lung cancer story, that's about 20% of lung cancer patients, that's a pretty decent number. But of course, what we're realizing now is we've found that low-hanging fruit. What we're finding now is molecular subtypes which are now sub 5%. But increasingly, that's still feasible to do. There's been a whole sort of paradigm change that many in the pharmaceutical industry are quite happy to now develop drugs which are now in 1 to 2% of a patient population if you've got a reasonably prevalent tumour. And the FDA is certainly keen to approve those. And again, that EML4 ALK fusion, does that make a difference? Well, this was one of the earlier studies. This is just over 80 patients, lung cancer patients, and they all have this EML4 ALK fusion. So this two genes stuck together which shouldn't be together. This is what's called a waterfall plot. So whenever you treat patients in a clinical trial setting, at the end of the trial, you look at whether the drugs worked or not and whether the tumour has shrunk. And essentially, the further your bar is below this axis, the better the response. So these patients are doing really well, and these patients really, you can see the tumour is actually increasing despite treatment. And we do a cutoff of 30% where the tumour needs to shrink by at least 30%. It's a very arbitrary cutoff, by the way. It's fascinating to read how they got to that 30% cutoff. It's sort of totally lick your finger and put it into the air. But this type of waterfall plot is a very good response. Most patients are responding to the drug, and it's another good example of targeting the right molecular abnormality with the right therapeutic modality. And you get these examples where this is a patient from that study. You can see the EML4 ALK rearranged lung cancer here pre-treatment, and you can see almost complete resolution. But not cure, of course, because again, in these patients, the tumour eventually comes back. In some of those patients, the tumour has now acquired a mutation in ALK. In others, it's not clear what's driving the resistance. But at least for these patients, you extend their survival for now. But we do have a huge piece of work to do around resistance, because we're beginning to get better at doing, if you cast your mind back to that patient where I showed you the melanoma patient pre-treatment, post-treatment and resistance, the first bit going from pre-treatment to post, we're doing that better now. We're defining more molecular subtypes, we're defining better ways to treat, but the resistance bit still needs quite a lot of work. And finally, and by no means least, mutational signatures. So everything I've talked about before, I think biologically is relatively straightforward to understand because it's about genes. Here's a gene. We know this gene has a function how cells divide and proliferate. This gene is genomically abnormal in some way, either it's mutated or amplified or it has a fusion. It's fairly self-evident that's driving something, and therefore if we target that process, the patients do better. But of course, it's possible to look at this genomic data and actually step back from the gene level and just look at the pattern of mutations. And the pattern of mutations in different parts of the genome, and also the pattern in which nucleotides are actually changed. And so if you could imagine that within any tumour, you have a whole set of processes which you capture. You're essentially capturing the archaeology of that patient from birth all the way to the tumour actually evolving. And at each step of her life, it's quite clear that cells in her body are exposed to different mutational processes. Those mutational processes are captured as an indelible imprint in our DNA, sort of archaeology. And normal cells will have these, but you can't always see them very well because they're not clonal. But tumours will have them as well. So tumours will have the imprint of many of the mutational processes that our normal bodies have undergone since we've been born, as well as those which are specific to the tumour. And so essentially you begin to look at these patterns, and if you look at every single possible nucleotide change So these are the, you look across the 4 nucleotides and the 6 different possible substitution changes. You begin to develop these imprints, and they're almost like fingerprints. And through looking at many thousands of genomes, there are probably, Florian can correct me, there's probably at least 30 or 40 mutational signatures now, and that sort of level. For many of them, it's not clear what the process is that caused that signature. And for some of them, it is. So, they're given numbers, and you can go into websites like the Sanger's Cosmic, and you can look at these mutational signatures, and they are quite subtle. But for example, if you're a breast cancer patient and you've got a BRCA mutant tumor, it has a very distinct signature. Signature 3 is a BRCA mutant signature, because that's a very specific defect in how DNA repairs itself in the DNA damage response in homologous recombination. This signature is from ultraviolet light exposure. So not surprisingly, if you have a skin melanoma cancer, you'll have lots of that signature. This signature, blues, bits of reds, bits of greens, is almost intrinsically linked with a condition called macrocyclic instability, which is quite prevalent in colon cancer and endometrial cancer and some gastric cancers. And again, this is a defect in a DNA repair process called mismatch repair, that if a cancer gets this, a defect in mismatch repair, then it can't fix the DNA mistakes, and it just accelerates really rapidly. And it has a very, very distinct signature as well. And the interest in these signatures is the extent to which, now, forget about what a gene does to survival and response, are signatures useful? And would we ever get to the point where, as well as using a gene mutation or amplification or fusion to decide which treatment a patient gets, would we get to the point where we actually use a mutational signature to potentially define how we treat a patient? And so the BRCA2 mutation in this particular signature is important because it's probably the paradigm for a condition called synthetic lethality, where you essentially have a pathway whereby you can't knock out both genes or the cell dies. And so what's clear in BRCA mutant tumors is essentially, if you can imagine, there's a pathway here where you need both of these feeding in to repair, both basic excision repair and homologous recombination. If you knock in homologous recombination, you still get the basic excision repair, so the cells can still fix themselves. Equally, we now have PARP inhibitors, but if you inhibit PARP, again, because you still get a functional BRCA, that's what happens. But you can imagine if you have a breast cancer or ovarian cancer which has a loss of function mutation in BRCA1 or BRCA2, this side's gone already. And of course, then if you use a PARP inhibitor to hit this side, then the cell dies. And that's really the classical paradigm for synthetic lethality, looking for loss of function events in specific pathways whereby the cancer, by having a mutation in one side of the pathway, actually leaves itself exposed as a vulnerability to targeting the other half with a drug. And again, one would argue now with the advent of CRISPR as a technology, so CRISPR is the ability to, with reasonably good fidelity that wasn't achievable before with short herp and RNAi, you can knock genes out or silence genes. There's been a few studies by this Sanger and the Broad Institutes knocking out every single gene across thousands of cancer cell, well, hundreds of cancer cell lines. I wish it was thousands. So we know which genes which knocked out on their own kill cancer cells. But of course now what people are beginning to publish are systems where you knock out 2 genes simultaneously. So you can set up libraries now where you start combining genes together for knockout. So I'm going to knock out A and B, or A and C, or A and D, and I'm going to work my way through a couple of thousand genes. You couldn't do the whole genome yet. And I'm going to look for genes that, when they're co-deleted, kill, breast cancer cells. And of course, then if you find that one of those genes that, when it's co-deleted, is also naturally deleted as a tumor suppressor gene, then you've got a synthetic lethality argument for a drug therapy. And I expect there'll be more of those. I mean, I talked about the fact that, when we're looking for the next generation of drugs, because CRISPR is such high fidelity and a very clean knockout, the next wave is going to be, I think, gene-gene knockouts for synthetic lethality and drug CRISPR combinations to see what deepens response to a drug treatment. So it's a pretty exciting time technologically. And again, is this important? So do you imagine that, if you look at patients with, BRCA mutations, you really do see, if you use a PARP inhibitor in these patients, they do much better. So these are patients who are have a BRCA mutation. They've got that BRCA mutational signature. If you treat them with olaparib, which is a PARP inhibitor, so that's second half of the pathway, then they do much better. They survive actually much better. And what's interesting in these patients is, whereas the previous studies this line eventually came down to here. They all eventually die. There are some durable responders. You do get some patients who do live quite a bit longer. You do also get resistance, by the way, even to PARP inhibitors. But you can begin to look across, tumor types, and this is, this is a study where you can look across every single tumor type and begin to define tumor type by tumor type how many have, a BRCA mutational signal, BRCA mutations, and you can see certainly prostate and breast. There's quite a lot, but you see it even ovary, some in colorectal. So I think this is a new way of nuclear molecular subtyping, and this is a paper from Serena Nix et al. Now using mutational signatures and looking. So if you imagine within each tumor type, and each of these columns is a tumor type, looking at these, 10 or 12 mutational signatures, and you can see here the proportion of each signature is indicated by the color scheme. These are the BRCA deficient mutational signatures, which are these two signatures here. So they really have a kind of a surfeit of those. And these patients potentially would respond to a PARP inhibitor. Now, what's interesting, and again, is that most of these patients who have a BRCA deficient mutational signature will have a mutation in BRCA1 or BRCA2, but they all don't have. There's quite a few who actually don't have mutation in BRCA1 and BRCA2. Some have mutations in other components of the DNA damage response, but some of them don't. So there's a question about if you treated these patients irrespective of the underlying gene event, but based on the mutational signature, will you get the same benefit with a PARP inhibitor? And again, what this shows is that, using new computational approaches, and this is 560 breast cancer samples from, again, Serena's paper. There's probably about sort of 91 patients who have out of that a high mutational signature. And again, the importance of this color scheme is you can see that, some of these in the yellow and blue have known BRCA or germline mutations, but there was about half were unknown. I think it's probably less than half now. But it's quite interesting. If I was that 52% with a very strong BRCA mutational signature, but the consultant was saying, well, you don't have a BRCA1 or 2 mutation, I'd probably be still keen to get a PARP inhibitor or a platinum agent, because we just don't know what's driving our signature yet, but it's highly likely, or at least it's a possibility. But that's quite a big question. And again, the same is true in pancreatic cancer, which is a very, very difficult to treat cancer with, almost universally a fatality rate of 100% in 5 years if you've got advanced disease. There's no real good treatment for pancreatic cancer yet. But, there are certainly a number of these. These are pancreatic cancer patients. This is a paper from Andrew Biankin. This is the BRCA mutational signature, and based upon a cutoff of, the propensity of the signature, the BRCA mutations per megabase of the genome, there were sort of, probably about 10 to 15% of the pancreatic cancer patients have a BRCA signature. And of course, within that, some do have frank BRCA1, BRCA2 mutations. So there's now an interest in certainly offering subsets of pancreatic cancer patients PARP inhibitors based upon the BRCA mutations. We're not treating at the moment, as far as I know, based upon mutational signatures. It's kind of a bit of a leap too far. There's still a bit of work to make sure that when we target the gene that's mutated, we get the right effect. But the mutational signature is an interesting premise. And I'll skip over that in the interest of time. So the second phenomenon I'll talk about is macroside instability. So this was this phenomenon where it's a very strong mutational signature. It's a bit like the BRCA mutational signature. It's because there's a defect in the DNA repair pathway. In this case, it's called mismatch repair. It's common in colorectal, endometrial, gastric cancer. You tend to get, all of us have scattered throughout our genome, essentially repeat DNA sequences. So we all have probably a couple 100,000 of these repeats throughout our genome. So we might have a run of all A's, a run of CA, 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 CAs, or even trinucleotide, dinucleotide, mononucleotide repeats. So we all have these. The problem is that they're entirely normal, but because they're repetitive, when cells divide, they're quite prone to picking up mistakes. And we're normally quite good at fixing these mistakes with the mismatch repair pathway. But if you've got a cancer which then has a defect in this pathway, then unfortunately these mistakes keep accumulating. And they're typically small insertions, deletions. And they give a very specific signature. So you can see, again, looking at NGS data, this is a colon cancer that was sequenced. You can see here, this is a mononucleotide repeat. Is it a dinucleotide? My eyesight's not, it's that AT, whatever it is. Oh, it's a mononucleotide. So that's a mononucleotide, and you can see here that there's deletions here just at the beginning of it. They're not on every single read because obviously most tumours have a degree of normal infiltrating cells as well. But that's what, if you're looking at NGS, that's essentially what you'd see in a macrosatellite unstable tumour. And they tend to have thousands upon thousands of these deletions throughout their genome. But what's interesting is that these MSI tumours are exquisitely sensitive to checkpoint inhibition. And I've talked about, targeted therapies, targeting genomic events, mutations is kind of really exciting and quite cool. Unarguably, the huge wave over the last couple of years has been immuno-oncology. There's negatives with that, by the way, in that too many trials chasing after a small number of patients isn't useful. But there's a phenomenon whereby certainly cancer cells, essentially, if you'd imagine a cancer cell chock full of mutations, many of those mutated proteins would get expressed in the cell surface, and arguably they should be detected by our T cells, and those cells should be eliminated. But there's a way that cancer cells are able to prevent that by essentially disrupting this interaction between PD-L1 and PD-1. And essentially what they do is they overexpress PD-L1 and essentially turn the T cells off and stop the cells being rejected. And so you can either block the PD-L1 here, or you can block the PD-1 on the tumor cell. But either way, you unmask the cancer cell to the immune system. And in cancers like melanoma, it's had a profound effect. Patients actually, I wouldn't say cure, but having very, very durable responses. But importantly, in tumours which have macrocyte instability, for reasons we do not understand, by the way, if you give these patients a checkpoint inhibitor, which is much less toxicity than chemo, they do very well. You can get patients who, again, get very durable responses and very high response rates. It's not licensed in the UK at the moment for this type of tumour. It certainly is in the US. But again, that's a good example. And so you can see the survival plots here. So if these are patients with colon cancer, treated with this drug, which is, again, one of these sort of immune checkpoint inhibitors. And you can see that if you've got macrocyte instability, this defect in this repair pathway, that's your survival here. Whereas if you don't, you're down here. And then I think, that's just how these patients get treated. So in colon cancer, this is probably only about 5% of patients with advanced colon cancer. It's also present in ovarian cancer, but again, it's another subset of patients where every colon cancer patient is no longer the same. We now routinely look in patients who have advanced colon cancer for whether they might have macrosatellite instability or defect and mismatch repair. Unfortunately, in the UK at the moment, we still have to go and ask on a compassionate use basis to the company to get access to the checkpoint inhibitors because it's not licensed yet. But that's done routinely now to offer those patients a different therapy, which is much less toxic than chemo, and more importantly, it is much better survival. And so, looking across, all MSI cancers, this is another one of those waterfall plots, and the more you are over to this side, the better. So you can see that in 86 patients who all had macrocyte instability, who all had this checkpoint inhibitor, the vast majority of patients did, certainly did better than you had of any other standard therapy. And I mentioned in the breast cancers that we're looking at BRCA deficiency, but interestingly, there were a couple of breast cancer patients who also have a macrocyte instability signature, but it's very, very rare. I mean, this was, quite a few hundred patients, and there were essentially a couple of them had MSI. But if you were a metastatic breast cancer patient, and I had a mutational signature of MSI, I would want a checkpoint inhibitor. But, we're sort of at the science stage where the science points in a direction we haven't done the clinical trials yet. And the clinical trials for this would be really, really difficult because you'd need to screen lots and lots of metastatic breast cancer patients to pick up these one or two. So that creates huge problems as to how we validate whether these things work in real life. And that's a challenge. So I've talked about how certainly genomics and coming from pre-genomic era has changed dramatically how we stratify patients and have a conversation with someone about how they should be treated, and the fact that not every cancer is the same. And so if you go through the FDA's web portal, you can actually track year by year which drugs they approved based upon a genomic biomarker. And essentially, it's just been going up and up. So it used to be in the old days, if I looked at those drugs approved, targeted therapies where you needed a genomic biomarker, It used to be I could get this easily on one slide. There were so few of them. It's now actually, it goes into 2 slides now, which is really cool. It has actually flattened out a bit. And there is this question about whether when we're looking at these genomic events, we may have found a lot of the low-hanging fruit. But it sort of has, certainly, this is up to 2017, so the number of sort of molecular markers which we used as a prerequisite, has gone up. Looking at this going out to 2019, it is beginning to plateau. And I talked about, the big waves now, the big challenges clinically, non-genetic resistance mechanisms via single-cell sequencing, defining, probably using CRISPR technology, more synthetic lethal interactions to add on to that PARP BRCA1 story. But I think while genomics has been a very powerful tool to identify druggable targets, I think we also now have to start thinking about how we drug in the non-genomic space. Classical non-genomic altered events, disrupted pathways, which may be aberrant not because of some mutational event, but maybe transcriptionally aberrant or epigenetically aberrant. And that's a very different set of biological models and a very different set of tools. So challenges, but quite exciting. Some quite interesting technologies. We've sort of learned a lot from this process. Life's getting more complicated because obviously we're now stratifying patients based upon genomic alterations, but those co-mutated events almost certainly have a role to play in, and we're still trying to tease out the effect they have. But again, just as a kind of an overview, as somebody who, and as some of us here, sufficient vintage that we've come from the pre-NGS era, we've come from a time where patients weren't stratified for treatment really in any meaningful way. Everyone was treated the same, by and large, by tissue type. To a time when, for those of you who are more recent, that probably seems unimaginable. Why would you treat an EGFR mutant lung cancer the same you treat one that has a RET translocation? But that is what happened. I think there are big challenges because we need to get away from perhaps a slight genome addiction. Not everything is DNA alterations to alter transcriptional programs, feedback, synthetic lethality. There's some big challenge, but also opportunities, and there's some really interesting technologies. I think, something that Flora and I discussed, that, for those of you who work in science at the moment, there are really, really nice technologies we have now that I think have revolutionized how we can look at different aspects of biology. I would make a plea that technologies are quite cool, but you should never let your technology drive your science. Think of your biological question, or your scientific question, and then pick your technology, because there is an element, some of technologies that are quite cool and sexy. People make those the centerpiece, and that should never be the case, because technologies come and go. It's really the question is the important bit. So again, I think this is the last couple of slides. So I would say we're doing this bit better, treating, not curing. We're stratifying patients much more effectively. We understand more about certainly the genomic alterations that predispose to better response to treatment. I don't think we understand particularly well the non-genomic events that do that. We're realizing that when drug resistance does happen, and it is inevitable, it is a sort of Darwinian process, that in some cases the drug resistance is druggable by adding in a second agent, but it is very heterogeneous. That's creating a big challenge in that if you have patients who are on a BRAF inhibitor and then there are 20 different resistance mechanisms, that means you need 20 different drugs. And you might need more than one drug for the same patient because people have biopsied different lesions in the same patient and you get different resistance events. And why would that surprise us? The metastases don't really talk to each other. They don't say, well, we're all going to go down the NRAS route. They're going to, depending on the tissue niche, they're going to adopt different resistance mechanisms. So certainly for the resistance side, there's a huge question about what happens to the, I hesitate to call it minimum residual disease because that's much more hematological term, but it is a sort of minimum residual disease. When you have these sort of almost a dormancy state here that's not genetic, that's much more plastic, that may have a metabolic as well as a transcriptional component, there's going to be a huge push to understand how these cells survive here that do eventually form the reservoir for the drug resistance. And that's a massive question, but a very, very exciting question for us to look at. And I'll skip over that. So I think most of this is self-evident. I think we're at quite an interesting time when we take for granted that patients are different. the molecular subtyping is different, and it's no longer sort of unusual to ask that we should extend that analogy. But it's not all genomic. I wish it was. It would make life a lot easier. I think there's going to be a very much a non-genomic phase of drug exploration coming up. And I will stop there, and apologies for running a little bit, not too much over. Thanks.
Okay, so this is called non-genomic resistance. How do you know that it's non-genomic, and what do you actually know about it? Are there any model systems, etc.? So
we don't know a lot. I think there've been probably a handful of studies where, typically in mice, where they've essentially sequenced, and many of these have been xenograft studies where they've essentially single-cell sequenced a tumor pre-treatment. They've treated, they've got a deep response. They've then collected cancer cells that survive the treatment, but in some mice they've continued treatment, so it eventually becomes resistant. So they have pre-treatment, the middle bit, and then the eventual resistance when it emerges. And by looking at the single-cell sequencing and also by looking at DNA sequencing, the late resistance is genetic, but the middle bit, they haven't found any obvious genetic lesions. But there's altered transcriptional programs which are biologically plausible that they're maintaining some type of resistance, and in some cases it's metabolic. But these are, handfuls of studies, and of course the old bulk sequencing a large amount of DNA, large amount of RNA, you would never be able to see these cells. It's really only with the advent of single-cell sequencing we can even look at what these cells are doing. Now, because it's possibly not just purely transcriptional, people are beginning now to layer on top of single-cell transcriptomics, metabolism, single-cell ATAC-seq. So I would imagine in the next couple of years there's going to be a lot more definitive studies, either in things like organoids or probably in mice, whether it's GEM models or PDXs, pre-post drug treatment using single-cell sequencing and a variety of different omics to say these cells that by definition survive treatment, because we know that if we continue treatment, if the classic experiment is you put your drug on, tumor regresses, you can't see it anymore. But then in some of the mice, you take the drug off and the tumor comes back. So there must be viable cells in that bit. And single cell means you now look at those. It's a big question mark, but probably sufficiently important to understand, because if the late genetic resistance was very homogeneous, then we wouldn't really worry. If we could target the late genetic resistance with 2 drugs, be an easy solution. What we're finding for most solid tumours certainly is the late genetic resistance is quite heterogeneous, and that's creating problems as to how we treat those patients. So understanding what happens in that middle bit, I think, is increasingly important.
But the fact that you get genetic resistance later on,
yeah,
that there's a huge benefit of that, right? And also the diversity in that suggests the same.
Yeah, so I think you're touching on a equally important question, which there is no answer to yet, which is how the non-genetic transcriptional programs eventually lead to the genetic resistance, and how that becomes hard-coded. Because at least in a couple of studies that have been done, that transcriptional non-genetic resistance, at least in the early stages, it is reversible. If you take the drug off, the cells flick back again. But what's quite clear is if you keep the drug treatment on for too long in the mouse, it becomes fixed. And we don't know what drives that. So there's lots and lots of interesting questions about not only what the programs are, but how they eventually become fixed. And how does that lead? Does that lead to increased genomic instability that eventually leads to the genetic resistance? We don't have answers to almost any of these questions yet. But we could at least begin looking now. But again, going back to have a biological question, and then we pick the right technologies for it. But there are some good technologies now to answer that. And it's a hugely important question, I think, because resistance is inevitable. It's in every single cancer patient. No matter how well we treat patients up front, they all get resistance eventually. It's almost inevitable in every single advanced patient, with very few exceptions, certainly in solid tumours. And it's quite difficult to treat. That's the problem.
I was waiting to see the students for that. Thanks, Ultan. That was a fantastic talk. it's more of I guess a philosophical question about, how much does targeting of signaling pathways really promise, and is there just too much plasticity in the system? And yes, the mechanisms of resistance to BRAF inhibition are heterogeneous, but they all converge on reactivation of MAP kinase pathway.
Yeah.
And then when we put the MEK inhibitors in, the patients do a little bit better.
Yeah.
And I would have thought that the resistance to that combination therapy would be activation of another pathway.
Yeah.
But it isn't. It's again just more gene dosage in the MAP kinase pathway. So,
but you do.
You see?
Yeah. So I go on the BRAF side, and so the kind of the BRAF MEK combination does better. I suppose the concern is that if we keep hammering the MAP kinase pathway, we'll eventually get something outside of the pathway. If you look at lung cancer, and EGFR mutant lung cancer, for example, it's very heterogeneous, that resistance. Even using our best EGFR inhibitors, there's probably about 20% of MET amplification, but after that, it's very small amounts of very different events. And that's very, very tricky for us to treat. So if you've got 20% MET, maybe 2-3% of a PIK3CA mutation, a couple of percent have a BRAF, a couple of percent have. And within each patient, it's entirely possible that different lesions could well have different events. And if you look at Roger Lowe's work, we did multiple metastatic sites in melanoma. They were all in MAP kinase pathway, but they were different. And why should that surprise us? So I certainly think we still need to we shouldn't stop defining the late genetic resistance, what happens, and whether it's druggable. I think we'll keep doing that, and we'll keep putting together those pie charts of the different slices on them, and trying to see whether there are any common segments that we would still drug and go after. We will keep doing that, and it's quite right to do that. I think there is quite an important question about what happens in between, and this kind of dormancy, minimal residual disease. What are those programs? Are they druggable? And they might not be, because of course the risk is that these cells that in many cases stop cycling, they may well be hijacking very conserved programs that would be very toxic if you tried to drug them. But it's just a massive question at the moment. So I suppose what I'd say is if you look at the two aspects of resistance, kind of late genetic resistance in the middle bit, we've been doing a lot in the late genetic space, and we'll continue doing that. But I think it's an opportunity to look at a slightly earlier stage. And I think that the kind of fairly sensible question is, should we be treating resistance earlier as a combination strategy? The challenge of combination strategies is we've all done it by putting together things we have in our fridge, rather than is there a systematic way to actually understand the biology that we want to target and use that to drive what we do. So if I was looking at a BRAF MEK plus something as a combination to stop that NRAS mutation happening a year later, can we do that systematically? I think, and again, we don't want to get into this, but I was at a systems biology talk, and the big consensus we all had when we're trying to put together, you talked about feedback and how pathways talk to each other. There's a huge need, I would say, for perturbational data sets, massive need. It's sort of crazy now that even for drugs that have been around for a decade, if you said, well, is there a panel of organoids or cell lines plus minus that drug with deep transcriptional omics data? In many cases, there isn't. It just seems quite bizarre. You can get into hand-waving what dose you use and what time point into that. But that perturbational data, I think, is something that we're beginning to build up within AstraZeneca. I think it's going to be an important part of how we define rational combination, understanding what cells do and how they adapt to treatments. And it is quite bizarre. There's just not a lot of it around. Arguably, that'd be quite a good resource if one was to take, whether it's with all the caveats about 2D cell lines or even organoids, if one was to take a very large numbers of these that covered many of the major cancer types, molecular subtypes, and did those type of perturbation experiments and made that into a public resource, I would imagine there'd be, computationally, non-trivial is how you then define pathways and networks and things, but difficult but not impossible. But that's a big gap in our knowledge at the moment. And again, when I talk about the kind of genomics addiction, we've got quite used to looking at a snapshot of a tumor fixed in time. That kind of longitudinal data, whether it's a cell line plus minus drug or a patient pre-post treatment, we just haven't been as good as getting those sort of data sets. But I think they are really, really important. Yeah. I've waffled a bit, I'm not sure.
Once we've mapped the feedback loops and the rest of it, it's possible that the conclusion will be that we can't.
I think we have to ask the question. It then goes back to another problem, which is to do that with biological models, we need more biological models. That there is a whole separate discussion is we're all using the same 5 cell lines to do every single piece of work, and that's not good. So, with organoids, we should probably be making a much richer set with more diversity of models. And the answer could be that we can't. The answer could be that there's so many different ways that cells can adapt that would be a 10-drug combination, and it would kill the patient.
There is a triple therapy in colorectal cancer, which is only, again, just gaining a few months. Likely is that we can't go beyond that.
Yeah, well, you do have. I mean, the
letalities are different.
Yeah, it is, and we're getting into kind of a toxicity discussion, which is really interesting because the BRAF-MEK combination was tolerated better than the BRAF, but that's unusual. I mean, but you can get up to 4-drug combination. There's a whole separate discussion from Peter Sorger's work about whether combinations really work by hitting different paths in the same cell, or whether you're actually each drug is hitting a different population of patients, which isn't as sexy, but it's actually maybe how a lot of these old chemo combinations work. We could any one of those 5 or 6 things we discussed there, we could talk about as a whole day. But I think we're beginning to realize where some of the missing data sets are, and part of the excitement I think from AstraZeneca coming to Cambridge is we try to work together to build some of those. Because what's interesting me in AstraZeneca is I came in expecting they'd be very much, oh no, we can't do anything with anybody, we've got to own everything, to very much in this space they're very happy to do a lot of stuff collaboratively. The competitive bit is the chemical molecule. This kind of stuff they're very relaxed about doing. So yeah.
Thank you for your talk. I would ask you to comment a little bit more on the role of the tumor microenvironment, just because we always focus on the tumor itself, but we forget the surrounding. So maybe we can just try to think that we don't need to target only the tumor, but also taking into account what happens in the surrounding environment, because it is very important. We know that it supports tumor evolution. So we start to think also about this, and maybe we can try to think about synergistic drug therapy.
Yeah. So a nice easy question. So you're quite right. And again, when I talk about this sort of the genome addiction, it's been very cell intrinsic, very cancer cell focused. And you're quite right. The tumor environment is massively important. I think the wave of immuno-oncology, but we haven't even touched upon macrophages, NK cells, cancer-associated fibroblasts. I think going back to what single-cell sequencing brings is the ability now to look at a population, and not just a population of cancer cells, but the entire population. And particularly if you use those longitudinally, then you can see how they wax and wane. I think, if the ideal scenario would be that we end up giving drugs that don't just hit the cancer cell, but maybe, for example, modulate NK cells, modulate T cells. In other words, we actually start drugging the tumor microenvironment. And arguably, some chemotherapies, that may be part of what they do. There is a lot of interest in that. It's quite difficult to model on the bench. That's, not impossible, but, as soon as people start setting up cancer cell, T cell co-cultures, NK, they are very reductionist by definition. So they are happening. People are beginning to look both on the bench and trying to modulate these. I think when you see papers getting published where people are running CRISPR screens in T cells, that's quite exciting, because for the first time, well, exciting for 2 reasons. Technically, the fact you can do it, you can now take a non-malignant population and actually turn off every single gene or large numbers of genes and look at some readout of function. That's pretty exciting. But what's even more interesting is the fact that people are thinking about doing it, because people are now moving away from the cancer and saying, well, okay, well, let's now look outside of the cancer. So I think it's just watching this space. I don't think there's any conclusive, sense of how we would drug the tumor microenvironment, how we would even assess a tumor microenvironment in a patient, in a biopsy a patient, how we would do that. But it's on everyone's radar. It's sort of a hot topic. It's not going to go away. There'll be a lot of interest in it. There's huge caveats about how you make those as preclinical biological models, how you look at them in mice, how you would look at them in patients, none of which are impossible. But it's the old George Box quote. All models are wrong, but some are useful. I mean, every model we use is wrong in different ways. I think the tumor microenvironment has a lot more complexity, and that's no different. Just stand up and say, well, here's my model. It's good for this. It's not so good for that. But that's another whole day's worth of drugging the tumor microenvironment. I mean, that's a whole conference. But yeah, really important. Yeah. So I didn't omit it because I don't think it's not important, but it's just impossible to cover in this time. But yeah.
Thank you very much. I know you have a busy day ahead of you,
yeah.
so let's thank Ultan again.